Hi dear students, now let's explore what is DDBJ, that is a DNA Data Bank of Japan. It contains annotated collection of publicly available nucleotide sequences, and it was started in 1984 at the National Institute of Genetics in Mishima, and it is even now being maintained. And you can see that in the website itself there are various options for us to either submit our sequence, or you can update your entries, you can do a blast search, or you can download FTP files, like that. Different DNA based analysis can be carried out using the DNA database of Japan. Now, it is mainly one of the centers which submit data to the International Nucleotide Database, Nucleotide Sequence Database. You have DDBJ EMBL as well as the GenBank, which has been provided by the NCBI. Three of these institutes together contribute towards the International Nucleotide Sequence Database. So, let's have a look onto it. Please come across, this is the DDBJ website, and you can see that they have different options. You can have a search or analysis, you can have a submission, you have downloads, and here the DDBJ, it mainly operates, and all the patents which have been submitted in the regions of Japan and Korea, or the nucleotide sequences as well as the amino acid sequence data related to the patent applications collected by patent office in Japan as well as in Korea, is mainly been submitted to the DDBJ, whereas all the data, let it be to Japan, Korea, Europe as well as USA, that comes into the INSD, or the International Nucleotide Sequence Database. So, let me just show you what are the features of this database. You can have a search and an analysis tool over here. You can submit your data, you can download certain data. You have a supercomputer here where you can process your files. You have a training which has been provided. Now, coming to the search and analysis, you have either you can search for annotated sequences, you can have a different sequence. You have a CRISPR, now CRISPR sequencing is being done, that can be done. You can go for a DFAST sequence, or you can have a BLAST sequence, different types, or you can go for a clustered W sequence. These are all advanced techniques which you can do, you will be learning in the coming sessions. So, you have different resources over here when you have to do a search or an analysis. If you want to do a submission, you can have annotated sequences submitted over here, or you can have from a mass submission system, different types of data can be submitted. Coming to the downloads, there are different downloads. Either you can have the 16 rRNA sequences, or whole genome sequence data. This is the supercomputer, NIG supercomputer system over here. Now, this is being shut down due to some maintenance. That's what the website is being provides this information. And you also have a center which provides you training, okay, either how to submit your data or how to annotate it. All this is being available in the DDBJ website. It's a database, so regional allocation of all the nucleotide based sequences which is from Korea as well as from Japan is mainly being given at DDBJ, and they share the information mainly into the the International Nucleotide Sequence Database. Now, let's see what is EMBL, or the European Molecular Biology Laboratory. It's a molecular biology laboratory which is supported by 27 member states, 2 prospect states and 2 associate member states. EMBL was created in 1974 and it is an intergovernmental organization funded by the public research money from its member states, and the database which has been maintained at EMBL is of much importance, as we mentioned, as a DDBJ. And the research at EMBL is conducted in approximately 85 independent groups covering the spectrum of molecular biology, and all the independent groups can be the list of these groups can be found in the website of embl.org, and you can see that the labs of the European Molecular Biology Laboratory is being located at 6 main sites the main laboratory in Heidelberg, and then you have outstations in Hingston as well as in Grenoble, Hamburg, Rome, as well as in Barcelona. And you can see that Israel is the only member state which is located outside Europe. So different laboratories in Europe are being linked together, and all their work is being deposited in a bioinformatics institute called the European Bioinformatics into the European Molecular Biology Laboratory. It is being as an EMBL website. It is being given. All the information of these nucleotides are being stored over there. So let's just have a look of the cute labs which are located at different places. EMBL at Heidelberg, and every lab at different location is having a specialization for different areas. Now here, if you go to Barcelona, they are specialized in tissue biology and disease modeling here. Here at Grenoble, it is mainly a center for structural biology studies, and here also in Hamburg also you have a structural biology studies. Like that, every lab is being specialized for different areas of research, but all the bioinformatic data which has been obtained from all these labs are being found in the EMBL website. I guess you get a chance to explore the labs over there. So if you go to conclude, I would like to say that all these databases which we explored today, let it be the GenBank is located in US in NCBI, the EMBL in Europe, or DDBJ in Japan, these are basically DNA sequencing databases or the primary databases where the raw DNA sequences are being deposited. So these are being found to be as primary databases. Now sometimes you can also have, as I told you, it is not, you cannot demarcate completely that a primary database should always contain raw data, because if you go to the GenBank itself, you can see sometimes some of these sequences are being found to be annotated. What is annotation? That is the function which has been linked with every DNA sequence is also been identified over here. And sometimes some of them will be completely fully annotated, so they are referred to as the annotated sequence databases. Or sometimes you could also have the low annotation sequence databases such as the EST databases.